Good afternoon. Welcome to Needham's 18th Annual Healthcare Conference. I'm Serge Belanger, the Spec Pharma Biopharma Analyst, and I'm happy to have these two gentlemen on the stage with me this afternoon, with Jon Stonehouse and Bill
Sheridan,
sorry. Who will tell us a little bit more about BioCryst and the important data set that we're expecting in the next couple of weeks, or the next couple months, sorry.
Do you want to start?
Sure.
Introduce yourself and then I'll go.
Yeah. Hi, everybody. I'm Bill Sheridan, the Chief Medical Officer at the company. Been with the company 11 years.
And I'm Jon Stonehouse, CEO of BioCryst, and I've been with the company over 12 years. And just to add a little bit more to what Serge said, it's a really exciting time at BioCryst. You know, we're approaching the readout of a pivotal phase 3 study this quarter. With success of that trial, we will file the drug by the end of the year for our first real commercial opportunity to launch in 2020. And alongside that, we're moving into a phase 3 study for the acute treatment of HAE attacks. We expect to start that study, a phase 3 study, in the summer. And we're advancing our earlier pipeline into the clinic, most importantly with 9930, which is a factor D inhibitor for complement-mediated diseases. So really exciting time at BioCryst.
Yeah. All right, so let's talk a little bit about the HAE market, where it's at. I know there's been a couple new products that were approved over the last 2 years, and it's really kind of regenerated growth in that market. And we can talk about the unmet need and where you think an oral product could play.
Sure. So let's look at the data. So the data at the end of the third quarter of last year says that the market now is $2.2 billion and growing at annual rate of around 20 to 30%. Currently, it's split pretty equally prophylaxis and acute treatment, but the trend is moving towards greater and greater prophylaxis, and we expect that will continue as a result of the introduction of more effective, more convenient prophylactic therapies. So the introduction of Haegarda, the concentrated C1 inhibitor given subcutaneously in 2017, the introduction of the antibody last year, and then our oral drug coming out next year. So we expect over time that about the time we launch, the market will be around $3 billion, and that the movement will be closer to 60-40 prophylaxis to acute.
Okay. And is that, obviously that move is from patients switching or beginning treatment on prophylactic treatments, or expanding in...
There is still a little bit of patient identification in this space in the U.S. Outside of the U.S., it's a bigger deal. But really, it's a switch market. And so they're either switching from one prophylactic to what they view as a better prophylactic, or probably more importantly, from on-demand therapy to prophylactic therapy.
Okay. All right, so tell us a little bit about your molecule, 7353. We've seen phase 2 data in APEX-1, and afterwards, I guess we'll definitely talk about APEX-2, the major piece of data that we're waiting for. But
Sure. So our molecule comes from our research group in Birmingham, Alabama. So it's homegrown. We have worldwide IP, no encumbrances. Composition of matter goes through 2035. Our goals were to have a long half-life, once-a-day oral medicine for prophylaxis in HAE. And those goals were certainly achieved in our phase 1 program, where we studied the PK and PD of the drug and the safety tolerability in healthy subjects. Then in the phase 2 trial, what we wanted to show was proof of concept in reducing the rate of attacks in patients with hereditary angioedema. So that was a 4-week study. Patients who entered that study were fairly severely affected with very frequent attacks, and the placebo attack rate on study was 1 attack a week, more or less. The outcome of the trial, and it was a dose-ranging study that studied 4 different dose levels, The outcome of the trial was we achieved all our objectives. The drug was generally safe and well tolerated. We confirmed the PK and the PD profile that we expected to get. We showed a no-effect dose at the lowest dose, which is useful from a regulatory perspective. 3 higher doses all worked, and we were able to pick doses to move forward into phase 3. So that's the short version of the story. I'm happy to say that that study got published in the New England Journal of Medicine last year, which is obviously a vote of confidence in the quality of the trial.
Yeah. And then I think there was some efficacy at some of the higher doses was somewhat impacted by GIAs. I don't know if you can say anything else about that aspect and how it can be mitigated in APEX-2?
Sure. So we learned in the phase 1 program that at the higher doses there was a low rate of generally mild, generally short-lived GI adverse events like soft stools or nausea or abdominal pain. This is a tolerability question, and in hereditary angioedema, what we learned in phase 2 in APEX-1 was that it can be hard for a patient with that disease to distinguish between a drug side effect of that nature or the early symptoms of an impending attack of angioedema in the small bowel, which could have similar symptoms. And the analyses that we did in conjunction with the PI of the study, and we showed some of that in the journal article, were pretty convincing that at the higher doses especially, subjects were confusing those types of symptoms with early signs of an attack. So when that gets recorded in the attack diary and counted as an attack, obviously that dilutes the treatment effect size. The key analysis that helped us come to that conclusion was to look at attacks that were purely peripheral, where there's swelling of the hand, for example, or the face or the foot, or attacks where there was a combination of that type of swelling in a peripheral organ plus some abdominal symptom. We identified those as mixed attacks versus events, at least, that were recorded with only abdominal symptoms and no other evidence of angioedema. And the higher doses had a very strong and dose-related effect on the peripheral and mixed attacks, but the reverse was true on these abdominal events. So it was pretty convincing. We wrote that up, and in phase 3, so in APEX-2, which is our phase 3 study, we've implemented a number of different tactics to minimize, first of all, the adverse event rate, and secondly, to minimize any confusion that might occur. So what are those tactics? Number one is dose selection. So we chose the dose that worked the best, and we interpolated a dose between that and the next highest dose on the grounds that we might get better efficacy, and it was worth studying. That's the first thing. So pick doses that are likely to have a lower incidence of those side effects. The second is to teach the subjects who come onto the study in the informed consent form and the investigators who come to the investigator meetings and run the study about this issue, so they're aware of it, because obviously we weren't aware of it prior to phase 2. The third is to have the site investigator, and this is probably the most important other than dose, is to have the site investigator contact the patient after each event that they record in their diary. So they have an electronic diary. Every day it asks, have you had an attack in the last day? If the answer is yes, and they press a button, that gets signaled to the site with the magic of the internet. The investigator in the next 2 business days approximately needs to pick up the phone, have a conversation with the patient, and ask questions around a standard set of concepts. And those concepts are, can you describe the symptoms? Did you treat it? What with? What was the response to the treatment? How long did the attack last? What's happened since then? Have you still got the symptoms? Is this similar or different to other events of that type that you've had in the past? Then the investigator makes a judgment on the basis of what they hear as to whether this is a real angioedema event or not. So investigator-assessed attacks that qualify and that are recorded by the investigator in the electronic case report form as angioedema events is what goes into the primary efficacy quantity that gets analyzed at the end of the day. So to give you an example, somebody might have had loose stools and a little bit of abdominal pain and said they had an attack. The investigator calls them, asks them about that, finds out about that, asked, did you treat it? Yes, I gave myself Firazyr. What happened? Nothing. I still have the symptoms, and it's a couple of days later. That's not an angioedema event in the small bowel, because it just doesn't work that way. So that ability to have that conversation was not present in phase 2. In phase 2, we had a remote committee looking at an Excel spreadsheet with a bunch of patient-recorded information. So that could help minimize any misattribution phenomenon.
And I guess when we compare APEX 1 to APEX 2 in terms of study design, clearly APEX 2 has more than 100 patients because you over-enrolled. Treatment's also out to 6 months rather than 4 weeks, and the patient population is also slightly different. It's going to be less severe patients than what you had in APEX 1. So how do you think of all these different changes and the potential impacts it could have on the efficacy?
The most important difference is really, and this drives the others, is the duration of the study. So you want to be able to show your drug works for longer than 4 weeks, obviously, and the standard in the field is about a 6-month study. So this is a 24-week trial. That's 6 regulatory months of 28 days, 6 times longer than phase 2. And in this type of disease where you're counting events that come and go, the analysis ultimately is the rate of those events, so it's a surrogate for the count. In phase 2, as you said, we had quite a severely ill patient population with an average of 1 attack a week on placebo. That's a small fraction of the HAE universe. You want to be able to show the drug works in a broader, more representative group. Obviously, you still have to have events in placebo, but in a 6 times longer study, you could afford to relax the criterion for minimum number of attacks quite a long way. We didn't go that far. We relaxed it to 1 every 28 days in the screening period. We expect, on the basis of past experience, usually you get a mean placebo attack rate of about twice the minimum screening attack rate. So maybe it'll be in the range of 2 attacks a month. That's plenty to show a difference between placebo and active on study.
And then the primary endpoint is a reduction in HAE attack rate?
Yes, the endpoint metric is the attack rate. The comparison is each active arm versus placebo on study. And the way the analysis is set up is with what's called a Hochberg step-up procedure. You run both analyses, you rank the p-values. If both of them are less than 0.05, you win on both arms. If one of them is greater than 0.05, the other one has to be less than 0.025 in order to win. So that's where the alpha is spent in that scenario.
Okay. So since it's obviously well statistically powered to show a difference of 20%?
So the original design had 32 patients per arm, much bigger, as you said, than the phase 2. That would have given us a power of 90% to show a 50% reduction compared to placebo. With the actual enrollment of 121 with about 40 patients per arm, that improves the power for that degree of reduction to 99%. So, the concept here is straightforward. Obviously, we could show a statistically significant improvement with less than a 50% difference under this circumstance.
Okay. And I guess the magic question, the one we get all the time is, what do you need to show? I think you were at 74% efficacy rate in a phase 2. What do you think you need to show in a phase 3 to be an impactful product in HAE prophylaxis?
Yeah, so whatever you thought about what drives preference, throw in the trash can in this disease, because what we found is it's completely different, and convenience and mode of administration drives everything. And that's not surprising because they've only had injectable medicines, and every time you stick yourself with a needle, every time you look in the refrigerator and see all the stuff, it's reminding you you're sick. If you're taking a single capsule a day, it's like your daily vitamin, you forget about your disease. So this idea of feeling normal is a huge, huge driver. So our belief is that there's going to be a really high demand to want to try it. And your call with Dr. Lumry last week is evidence of that when you asked him how many patients do you think want an oral? And I think he used a number of 85%, even after he just described to you how well they're doing on other therapies. And that's consistent with the market research we just conducted in November of last year. That's consistent with the market research we did in 2017, where we varied efficacy from 55 to 85 and saw very little change in preference. So I don't know the exact number. If we get a statistically significant result that we think is clinically meaningful, and remember, those are means. That, what matters is how each individual patient does. And so if an individual patient has most of their attacks or all of their attacks are gone, they're going to love this therapy, right? So I believe our job is to make sure that patients stay on our medicine and make it as convenient for them to do that as possible.
So trial reads out in the second quarter?
Second quarter. Everybody's been trying to figure out when, but it's second quarter.
This quarter. And there's an extension study. I mean, can you just walk us through,
Sure.
Once you get the data, what are the next steps to filing an NDA and so on?
So the pillars of approval are safety, efficacy, and quality. So the efficacy is the 24-week readout and a strong contribution to safety. That's really the critical safety information compared to placebo. But there's also a longer-term safety evaluation. Patients roll over into a 24-week extension, and we also have an additional open-label safety study to augment the numbers. So between those two studies, we need 100 patients at each dose level to file the NDA through 48 weeks total of observation with active drug. So that won't happen until later this year. That's the rate-limiting step for filing an NDA. We're confident we should be able to hit that this year and file by the end of the year.
So gating items is really just clinical data. It's not product patch stability.
No. In fact, on that front, we're in fantastic shape. So we've got a second supplier for both drug substance and drug product to just make sure that if something goes wrong with one, we've got another in the packet and we can still get approved. We're already starting to write up the CMC sections of the NDA module for that section. We're writing up some of the preclinical sections as well. So we're absolutely on track, and with the data coming from long-term safety, that is the gating item.
And how do you think of commercialization for this product? I guess in terms of sales force sizing and pricing, I thought one of the interesting comments from Dr. Lumry last week was potentially pricing an oral at a discount and forcing basically payers and forcing patients to step through a cheaper or more affordable oral product before going on to the injectables.
Yeah, so the beauty of rare disease and the beauty of having an oral small molecule is everything is less expensive, right? So commercialization, sales force, this isn't the exact number, but you can think around 40 people instead of 1,000 people in a general market. A handful of MSLs to support the drug, and so the investment is reasonably low, and then with the cost of goods with a small molecule, you have a lot of flexibility. I can't answer you specifically on how we think about pricing yet because I don't have the profile as a result of the APEX-2 trial, but having that flexibility is fantastic, and we'll make sure that we get the best value for the profile that we produce.
And then thinking about ex-US plans, I know there's a Japanese trial ongoing or it was ongoing. I guess just what are your plans in terms for advancing or filing the products in either Europe and Japan?
Yeah, so we just added a couple of new board members to our board, Teresa Heggie from Alnylam and Steve Asalegi, who used to be the CEO of Retrophin. And both of these folks have really extensive rare disease commercial experience. Teresa runs the European group and some other territories at Alnylam. And so we're getting some really good advice. And the good news is we retain all the rights. So Europe, we plan to launch ourselves. And we're putting together plans on, the hiring and the buildup of that. We expect to file in Europe shortly after we file in the U.S., so in the early part of next year. We'll do that in Japan. I just got back from Tokyo late last week, and we're exploring both You know, is it better to put it in the hands of somebody else, or is it, with rare disease, easy enough where with, 10 reps you can cover the whole country and manage that? So we're exploring both of those as well, and we'll make a decision sometime later this year on that front. But again, holding the worldwide rights is of great value.
Is it disease that's different in the Japanese population? Do you think the drug acts differently?
Well, the disease isn't different, but the diagnosis appears to be a lot different. So in Japan, it appears that they're about 10 years behind Europe and the U.S. in terms of identifying and diagnosing and then approving drugs for the disease. So right now, all that's available is Berinert, and Firazyr was approved just recently. And so there's no prophylactic therapy approved. We have Sakigake designation. As you said, we've got a small trial running in parallel to our pivotal APEX-2 study. We'll combine that data with the pivotal study and submit to the Japanese authorities. And so there's a real strong interest in our drug by the authorities and by the patient association and physicians who treat these patients. So I'm hopeful that other companies will do some of the heavy lifting in diagnosing the patients, but if we have to do some of that when we get to market, we'll do that as well.
I guess speaking of the other companies, Shire has always been the major player for NHAE. Have you noticed anything since they've fallen under the umbrella of Takeda?
I think they're doing a good job. My sense, having been at QuadAI, we saw a pretty sizable presence at that meeting, but having been through some pretty hefty mergers in my career, there's a distraction factor and a loss of talent that sometimes happens with that, and we'll see if that impacts. I haven't seen any evidence of that yet, but that's certainly a possibility.
I guess moving on to the other 7353 program for acute HAE treatment. So you're using the same molecule that was designed to have a long half-life. Obviously, you made some formulation changes to it. Can you just talk about what those formulation changes do to the PK profile and what kind of onset you see and stuff like that?
Sure. So the long half-life turns out to be a tremendous advantage for acute treatment as well, because with a single dose you can cover the target enzyme for longer than 24 hours. With the dose that we've selected to take forward into phase 3, we get more than the upper limit of normal coverage of the enzyme within an hour that lasts right through 24 hours and beyond. So the likelihood that second dosing will be needed for an individual attack is extremely small with this compound. So that's a useful characteristic. The objective with the formulation development program was to have something that would dissolve quickly. We didn't have that ready for phase 2, so we gave the drug in a liquid form. That's not ideal for commercialization with small molecules because of stability issues, plus the taste issues of most small molecule drugs. So the phase 3 and commercial formulation of the drug is a very clever mini-tab formulation. They're about 3 millimeters on a side, and a small volume of those constitutes the dose. Tasteless completely, can be swallowed simply with water, and it's a pH-sensitive coating. So when it hits the stomach, it instantly dissolves. So it's very similar in that regard to giving a liquid, right? So it's very important also to note that the dose, because it's got such a long half-life, in order to get the coverage we need, the individual dose for an acute attack used for rescue treatment has to be higher than the daily maintenance dose when you're at steady state with the compound. So that's why the dose is 750 milligrams. But it's exciting. I think the phase 2 was a very well-designed, novel program. Historically, this field has been very difficult to run studies in and win in the first step into a development program. So that's an achievement. We look forward to completing the regulatory interaction soon and starting the phase 3 in the summer.
I think it's worth just expanding a little bit on what Bill said, because I think there's some confusion around what profile do you need to have an effective therapy in this space, especially when we've moved from treating patients in the hospital or the clinic hours after their symptoms start. So what does the data tell us? The data says that you need to get to this threshold and maybe a little bit higher, and you got to get there quickly. and we show a clinically significant difference at the first time point we measure, which is an hour after they've taken the drug. So if we measured it earlier, maybe we'd see it even faster, but an hour is pretty darn fast. And remember that their visual analog score, their baseline score is 14. So how are you going to improve dramatically on such a low score out of 100 points to begin with? So that's number one. So this idea that you got to just rock it, I think there's no data to support that. The second point is that we're now starting to see data come out, both in a study that was done in Berlin and another study or poster that was presented at Quad AI by the HAE Association, that the real redose rate of short half-life drugs is high, 30 to 40%. If you're a payer, the last thing you want to do is pay for a second dose. If you're a patient, the last thing you want is to take a dose and go, well, is that enough or do I need to take a second one? So it has to go to work fast, but you don't need crazy amounts. Get above the threshold, and you got to work for a long period of time. That's the profile that we think is important in the acute setting.
And I think the main questions around this program is, what will the phase 3 endpoints be? And I guess we really won't know until you meet with the FDA and agree on them. But from what the endpoints you looked at in the phase 2, ZENITH-1, which ones do you think are more impactful or most likely to be in the phase 3? Some were questionnaire-based, some were more pain-based.
Let me take this because I want to toot his horn a minute. So one of the brilliant things that Bill and his team did is they put in a bunch of endpoints in different categories: use of rescue medicine, visual analog score, survey questions on global assessment. And I can't remember the total number that was put in, a dozen maybe. And 2/3 of them we have statistical significance and they're meaningful. So what that gives us, and this is where I think they did a great job, is choice and flexibility depending on what direction the FDA takes us. So I'm confident that we will come up with an endpoint that we can all agree upon with the regulators.
And then has the phase 2 meeting been scheduled at this point, or still?
We haven't given that kind of specifics. What we have said is that we'd like to start the study this summer. I've been guiding today. It's probably closer to the back half of the summer, but the goal is to get it up and running this summer.
And what would you expect to see in terms of size, number of patients, and things like that?
So the division we're working with at the agency is fond of parallel group studies. Okay. And you can take an example from the migraine literature and the way those studies are done. It's another episodic disease. So the way those studies are done are randomizing subjects to a placebo or active for their first event that happens after that. Then after the first event, then it's just a safety collection in open label. So that's a parallel group design. The studies could be quite large in the rare disease context of 80 or 100 patients per arm. That hasn't been settled yet. And I think that at the moment, in the absence of having finalized the design, it could take a year or 18 months to run through all of the subject recruitment and get the events you need to do the analysis.
Okay. And I know you've done extensive market research in both the acute and prophylactic segments. Do you think orals have a bigger role to play in either of those segments, or it doesn't matter, they'll be welcomed really in both?
I think the interest level is really high in both. I mean, we remember how this all unfolded. We had the prophylactic program first, and that was the only program that we were pursuing, and patients and physicians told us we should study it in acute as well because they believed that it had an interesting profile. I can't tell you the number of times Bill and I get the question, when are you going to study the two together so we have an all-oral option? But in the market research on the acute program, one of the things they told us is, if you've got a formulation like our Minitab, I'll probably treat myself earlier than I would have with an injectable. So that tells you something as well. So I think it's applicable to both.
And then, obviously from the market research you've seen, the high demand is attracting other players to the space. Could you give any comments on some of the competing programs that are out there and
Yeah, it's just
either orals or some, I think gene
just,
therapy is also being looked at and things like that.
Yeah, I think it's just harder if you're coming in second and you're behind. You got to have a profile that's better if we get to market, and that's hard when you got a once-a-day oral prophylaxis. Finding and making a kallikrein inhibitor that has a half-life like ours, we're finding out is not so easy, right? And then if you have an acute therapy and you're a couple years behind, again, you got to be better. And then gene therapy, one of the people from the Patient Association said to me, why would we consider going into the risk of a gene therapy study when we have drugs now that basically eliminate all of our attacks? So that says enough for me to, not be as worried about gene therapy in this space.
Okay, so we should see an NDA filing by the end of this year. How do you think about the commercialization build up for this program? Is this something that takes place over 2020, or you start looking at it?
No, we're starting now. So I mean, the number of interviews that are going on in our company and the number of offer letters that I'm signing, I think we're now up to at least 100 people in the company and growing. And the idea is that we build the commercial medical affairs. We have, you've met Lynn before, our chief commercial officer, but she's got a bigger team than the last time you saw her. And Bill has hired a head of medical affairs. He's in the process of building out the medical science liaison team. So yeah, we're gearing up now. The sales force we won't hire until we get closer to approval, but the plans for that, as you said, we're trying to get smarter than anybody in terms of understanding how switches happen. That's one of the great things of watching what's going on in the marketplace now, is we can gather data on that. So we're very, very active in getting ready for launch. We're not waiting.
So we only have a couple minutes here left. Mind talking about the new pipeline program?
Love to do that.
9930.
So, Factor D is a number one target for treatment of complement-mediated diseases. Our drug is an oral Factor D inhibitor. We've got a lovely pharmacodynamic profile of suppression of the alternative pathway of complement in non-human primates in our preclinical development program. Our safety margin is huge. The human equivalent dose of the NOEL is 5 grams. We'll never need to dose that high, but it'll give us a lot of flexibility in the clinic. So that's moving into phase 1 SAD/MAD this quarter, and by the end of the year we'll have safety, tolerability, PK, and very importantly, the effects on the complement pathway in healthy subjects by the end of the year. So a beauty of complement as a field is that there are commercially available assays, and you can show effects in healthy subjects that will highly likely be very predictive of benefit in patients.
And there's a whole slew of complement diseases. How do you choose which ones
all of them
to go after?
All of them. So the strategy is really straightforward. We don't want to tip our hand to the competition because, as you know, it's crowded. But the strategy is do the SAD/MAD, then do a proof of concept study in one indication. These are small studies. You get results in a couple of weeks, and you can test a handful of patients. Once we've shown that, go in a bunch of different diseases with the same kind of proof of concept studies in parallel, and then run the pivotal program for that lead indication. And so, that could all start next year. And so you think about filing an HAE drug, getting approval and launching an HAE drug, and then having a pipeline in one molecule with 9930, the value of this company starts to get really attractive.
We only have a few seconds left here. I know none of you guys are the CFO, but does one of you mind running through the financials?
Sure. So we ended the year with $128 million in cash. We then refinanced the loan with MidCap to add another $20 million in cash, so that put us around $150. We have another $30 million that we can draw with successful data from the APEX-2 study, so in the second quarter we could draw that. And our burn rate is somewhere between 105 and 130. So what that mid-cap refinancing does is it just gives us extra flexibility rather than having to immediately raise cash. Not saying we won't do that, but it gives us flexibility.
Guess we'll stop there, and we'll wait for important data over the next couple months.
Thanks, Serge.
Thanks.